Everyone, welcome to my YouTube channel, and today I am going to give you a brief process on DNA extraction, on which I will deal about the principles, laboratory protocols, and the process for isolation of high purity of DNA. My name is Prakash Bajagain, and I have completed my master's in microbiology along with the bioprocess engineering from France. Without delay, uh, objective of DNA extraction. The reason behind to extract DNA includes to obtain a DNA in its pure state from its complex sample, either from plants, animals, or even a prokaryote and other eukaryotes. To extract DNA, initially we need to remove the contaminants present in the cell. The contaminants in the cell includes while targeting DNA like a protein, RNA, lipids, polysaccharide, which should be removed from the solution. To maintain the DNA integrity, it is necessary to do PCR, sequencing, and cloning. For this purpose as well, DNA need to be pure and highly quality. General workflow over the DNA extraction includes cell lysis, on which initially cell, depending upon the morphology, it is necessary to break their wall and the membrane to make DNA efficient to release. So it is necessary to remove the unwanted proteins uh, in these cells to get DNA. So it is necessary to do precipitation or digestion of a protein present on the cell. For that, um, we have to do protein removal, and to get a DNA in the form of precipitate, DNA need to be make insoluble. For that, we can do alcohol treatment. Cleaning and hydration of DNA. For that purpose, we have to remove the salts present on a DNA and suspend them in a TE buffer. Now, I like to let you know in depth cell lysis and destruction of a DNA. For that, we can either do with mechanical, on which we go with grinding with the like mortar or pestle, or through the bed beating techniques, or like a chemical method on which we can use sodium decyl sulfate to disrupt the lipid bilayer, and also we can use CTAB method for the plant DNA extraction. Behind that, like in case of prokaryotes, we can extract DNA with the enzymatic hydrolysis, on which we can use proteins which will be able to destroy the unwanted cell wall of prokaryotes. For the purification of DNA and just to separate its phase, initial stage includes extraction of pure form of DNA using phenol chloroform method. So on putting phenol and chloroform mixture in the lyased DNA, it is necessary to separate with centrifugation, which separate cell based on its density. In the aqueous layer, there will be a DNA, and in the interface, there would be the protein, and organic debris layer will be at the bottom of the centrifuge tube. For the precipitation of DNA in insoluble form, it is necessary to do treatment with alcohol and It is possible to use cold isopropanol ethanol for the precipitation, and for the neutralize charge of DNA we can use salt like sodium acetate, and through the precipitation DNA will appear white in color and thread-like structure. With that appearance we can identify, oh, we have precipitated DNA. So final washing and elution includes 70% ethanol treatment, which removes the salts present in the DNA precipitate, and also will help to air dry. We can do air dry to pallet to remove the ethanol, and we can load them in tea vapor or the water which is nucleus free. And to check the quantity of DNA we can use spectrophotometry, especially ultraviolet. For the purity of DNA we can check with like again spectrophotometry at the 260 nanometer and at 280 nanometer. If DNA having strength of 1.8, it's count to be pure DNA. And for the quality to test, aerogel electrophoresis will plays a vital role. Thank you so much for your time and attention. In next video I will let you know about the PCR technique in detail.